All right, welcome everybody to Standing for Truth. My name is Donny Budinsky, and I am your host for today's exciting program. On Standing for Truth, we defend the truth of biblical creation. We also host some awesome guests on the program. And today, as always, it is a privilege to have Dr. Peter Line with me for this very important show. Now, today, Peter, a neuroscientist, is going to dismantle human evolution by looking at the incredibly designed human brain. This is the second part of Peter's must-watch presentation, and I am excited for what he has for us on this topic. Now, before we get into the second part of this presentation, I do want to give Peter Line a formal introduction for anybody who might be new to the channel and maybe unfamiliar with who Peter is and his work. Peter's undergraduate majors were in biophysics and instrumental science. After this, he completed master's and Ph.D. degrees in the area of neuroscience, focusing on brain electrophysiology, and spent over a decade involved in such research. Subsequently, he worked as a university lecturer, teaching in the area of anatomy and physiology for over a decade. Peter has been interested in the creation/evolution issue ever since becoming a Christian, as evolution was a stumbling block for him, believing God's word was true. He had a particular interest in the so-called hominid fossils for many years, and has published several articles on the subject. He's also done a 3-part, incredibly comprehensive series on the hominid fossil record and the icons of human evolution. So for those, he's done that here. And so please, for those who have not yet seen that series, do check the website, standingfortruthministries.com. And I've also got those shows linked in the description box of this video. And so, Peter, my brother, it's great to have you as always. How are you doing today?
I'm pretty good. Thanks, Donny. Yeah. So
okay. Well, let's get right into it then. I'll let the audience know as you're presenting. Peter, I'll be taking audience questions and challenges. Once you're done your presentation, we'll spend a little bit of time engaging and interacting with those, and then we'll wrap things up. So, Peter, I'm going to hand it to you, brother. I've got your slides up, and the floor is yours.
all right, thanks very much. Yeah, so unfortunately last time we got kind of cut off, and so I'm going to start off with the slide before the one we got cut off, which is about, we were talking about the spinal cord, so that people can follow from the start of at least this part. Okay, so we're looking at the sectional anatomy of the spinal cord, and basically you have each spinal segment receives incoming, that is called afferent or sensory information, from what's known as the dorsal roots. Here you can see these things here. So they're basically axons of neurons from dorsal root ganglia, shown here. Like, dorsal root ganglia are a collection of cell bodies of unipolar neurons. Okay, and ganglia is a name given to a collection of neuron cell bodies which are outside the central nervous system. So basically, the sensory connection tend to project into the dorsal or posterior section. Sometimes it's an unfortunate thing, but in a spinal cord, or basically ventral is sometimes called anterior and vice versa, and dorsal is sometimes called posterior and vice versa. So you have these kind of different names given to describe the same thing, but yeah. So, but anterior ventral is the front of the spinal cord, posterior dorsal is the back of the spinal cord. Now, so now you also have a pair of ventral roots shown here. That contains outgoing information in the form of axons of motor neurons, which extends into the periphery to control somatic and visceral effectors. So an effector is basically a peripheral gland or muscle cell innervated by a motor neuron. Hence, basically, motor connections project out from the ventral or anterior section of the spinal cord. Okay, and I think this is a slide which kind of got cut off last time. Okay, so you have here the gray matter in the spinal cord is divided into laminar or divisions, often called Rexard's lamina, and consist of 10 layers. And they're divided in the basis of architecture and longitudinal organization. Now, they tend to run parallel to the long axis of the spinal cord. And these layers form part of either the dorsal, that is posterior, or ventral, anterior gray horns. And basically, these regions, so it's dorsal or here, ventral gray horns. Now, part of layer 7 is sometimes referred to, shown here, is sometimes referred to as a lateral gray horn. It's only present in thoracic and lumbar segments, so that contains visceral motor neurons. So they are basically ones that visceral or relates to sort of autonomic nervous system functions. Now, the dorsal gray horn, or up here, posterior or posterior horn, is formed by lamina 1 to 6 here, and these contain sensory nuclei and process incoming sensory information, particularly about pain, temperature, touch, itch, as well as limb position sensors. The ventral gray horn or anterior horn, sort of, get my cursor back. Where is it? Hang on. Here. So here is formed by lamina 7 to 9, and of the spinal gray matter. And these lamina contain motor nuclei, and they're shaped more like rods or columns than the flattened sheets, laminated sheets of the dorsal horns. And the lamina X comprises gray matter which surrounds the central canal of here, and it's often referred to as central gray, and it's kind of involved in autonomic nervous system functions. Okay. Now, the white matter on each side of the spinal cord can divide into 3 regions called columns or funiculi. So you have the posterior or dorsal white column. Sometimes you call them white, but well, usually you just omit the term white. You just call them either posterior or dorsal column. Then you have the anterior or ventral column, and then you have the lateral column here. Now, these columns contain tracks whose axons relay the same type of information, whether motor or sensory, in the same direction. The longer tracks connect the spinal cord with the brain, whereas the shorter tracks carry signals, whether sensory or motor, between segments of the spinal cord. So the posterior column contains ascending axons of mechanoreceptors, which is sensitive to mechanical stimulation, for example, touch, vibration, pressure. The anterior column here contains primarily descending motor fibers for controlling axial, that is head, neck, and trunk region, and also proximal limb structures. And then you have the lateral column, contains here, in this region here, basically contains diverse somatic, sensory, cerebellar, and motor control pathways, including more precise movements of distal parts of the limbs. Let's get my next slide. Now, the posterior or dorsal column pathway delivers fine. So basically, you're getting this posterior, the green bit at the back here, delivers fine touch, vibration, pressure, and proprioception information to the primary sensory cortex on the opposite side of the body. Proprioception means that sort of positioning, relative positioning in space, or things like that. Now, ascending axons from the posterior column synapse with dorsal column nuclei here. The nuclei gracilis and nuclei conatus. So you have these axons that project into the spinal cord. They move up the posterior or dorsal column, synapse in these nuclei here in the medulla, and then basically they project ventrally, sort of forwardly project through the medulla, where they call the internal acute fibers here, and then they basically enter a tract. The tract is then called the medial lemniscus, which then moves on to projects up to the thalamus. So these neurons synapse on the thalamus here. Now, the lateral spinothalamic tract, which is, I'm just getting my cursor back here. It's a bit awkward, kind of moving it back and forth here. This tract here carries sensation of pain, and temperature to the primary sensory cortex on the opposite side of the body. So it basically, instead of going up the posterior column here, it synapses on the gray matter in the spinal cord, projects to the lateral opposite side here, and then just goes as a lateral spinothalamic tract right up to the thalamus here. Okay. And now I'll just to the next slide. And so just looking at the corticospinal pathways, so that's the motor pathways. So basically the axons in the corticospinal tracts located in both the anterior and lateral columns synapse on lower motor neurons. Hang on, here we are here. So these basically synapse here on lower motor neurons in the spinal cord, in the ventral gray horn area here. With respect to the corticospinal pathway, as the axons of the upper motor nerves of the primary motor cortex descend here, labeled as cerebral peduncles, most of them, about 85% of them, cross over here in the lower medulla here. 85% of them cross over, and then descend as a lateral corticospinal tract here, and then gets to the gray matter here, and then it synapses here on the gray matter. So that, and then the motor nuclei is within the gray horn, so that's part of the central nervous system. But the projecting axon, once it leaves the CNS, or then it becomes part of the peripheral nervous system. And you also have the other part here, about 15% or something goes to the anterior corticospinal tract, which then goes straight down, and then to the area of the spinal cord, and then it crosses over here, and then crosses the other side here, and then synapses here on the motor nuclei down there. Now, just trying to get my cursor back here. Okay. Now, the above shows how information from the motor system is relayed to the skeletal muscles. Again, it is all very orderly. I think that is the point I'm trying to convey. Now, white matter that is myelinated nerve fibers make up a considerable portion of the nervous system, connecting numerous different regions in a very orderly way. It's actually been calculated, if you stretch out all the nerve fibers in the human brain, this white matter, they would wrap around the globe 4 times. Okay, that's not me calculating, that's evolutionists calculating it. Okay, so how did this order arise? Without intelligent planning. If it was created by a purposeless process such as evolution, if it were possible, then one would expect more of a spaghetti and meatballs arrangement, not orderly parallel nerve fibers and tracts, with nerve fibers making up the individual tracts carrying similar information. Now, so let's have a look at the sensory homunculus. Now, as the cerebral hemispheres must carry out many functions, some type of intrinsic organization is necessary. Somatotopy is the orderly mapping of the somatic sensory system and motor system on the cerebral cortex. This organization is by function and by body area. Now, the homunculus is a schematic representation of the cortical projections for motor and sensory functions, where the body is drawn in proportions to the amount of cortical tissues that subsumes each part. For example, the sensory homunculus shown above has a prominent tongue, face, and hand, including fingers, and a relatively small trunk, hip, and leg. Now, these proportions correspond to sensory discrimination. That is, the density of receptors is important. The greater the density, the more cortical tissue needed. The size of the body parts is less important than the density of receptors in determining how much space is needed in the cortex. Now, for the motor homunculus shown here, on the left, above left, it has a large face, tongue with respect to speech and swallowing, and large hand and a relatively small trunk and hip. Now, with respect to motor control, the innervation ratio of these regions is important, which is the number of muscle fibers which are innervated by one motor neuron. So a lower innervation ratio allows finer motor control of movements and requires more cortical tissue. Think of um, a puppet on a string, where if you only have one string, you can't do you can't really move it much. If you have dozens of strings, you can really do fine movements, and that's a bit like that. Now, the homunculus shows what a body will look like if each part grew in proportion to the area of the cortex of the brain concerned with its movement, that is motor or sensory perception. Now, so and representation of the cortex is of the contralateral, that is the opposite side body parts. As mentioned, the cerebral hemispheres must carry out many functions, and so some type of intrinsic organization is necessary, or at least useful, and that is what is seen. But is this really what you would expect if it wasn't designed? Remember, evolution does not involve any planned design, involves no intelligence, does not see ahead, and does not care, or in common vernacular, give a hoot. It supposedly makes do with what it has to work with. Notice the many similarities in the orbit of the body parts of the primary somatic motor and somatic sensory areas, which are situated on a precentral and postcentral gyri, respectively. Is it just coincidence that this is so? The probability of this being due to luck is very low. If the evolutionary process actually was responsible, you would expect a more disorganized body parts assembly in a way it is represented on the cortex in terms of the order, and not just limited to the precentral or postcentral gyri. And you would not expect such a close correlation in the order of the body parts of the primary somatic motor and somatic sensory areas. The above organization has all the hallmarks of being designed by a super intelligent engineer, not by something decided by nobody that is nothing without plan, intelligence, and purpose. Okay, now let's get on to vision. Okay, now concerning vision, consider the above statement by international award-winning author James Lefanu. He says it had long been supposed, for instance, that the visual cortex at the back of the brain acts as a sort of photographic plate capturing an image of the external world as seen through the eye. But now it turned out that the brain created that image from the interaction of 30 or more maps within the visual cortex, each dedicated to one or other aspect of the visual image, the shapes, the color, movement of the world out there. As surely as the old system was rooted in the concept of an image of the visual world received and analyzed by the cortex, observed Zemel Seki, professor of neurobiology at the University of London, the present one is rooted in the belief that an image of the visual world is actively constructed by the cerebral cortex. Also, according to Jerome Feldman, one of the most famous continuing questions in computational neuroscience is called the binding problem. In its most general form, the binding problem concerns how items that are coded by distinct brain circuits can be combined for perception, decision, and action. For example, the visual binding problem has to do with why don't we confuse, for example, a red circle and a blue square with a blue circle and a red square. The above is how the problem is studied in a Neuroscientist publication. It says, we now know that while different tasks are carried out by different cortical regions, for instance, these tasks are combined smoothly. Depth, shape, color, and motion all merge into a 3D image of the scene. For example, we recognize objects with no awareness of the fragmented nature of the brain's efforts. Precisely how this is achieved is called the problem of binding and is a challenge for the next generation of scientists. Okay, this shows the parcellation or division up of different cortical regions involved in visual processing. So on the left, you can see here you have the look as a side lateral side view. It shows the uh, temporal region and the right temporal region and the occipital cortex, and the right side image is a inferior or ventral view of the brain. Okay, remember when well, I shouldn't say remember, but in with the brain, because the neuraxis or longitudinal axis it bends around the midbrain, what is ventral anterior becomes inferior in the brain because it bends. So that's why I say ventral. Um, but it's so it's actually better just to use inferior because it just is more confusing in terms of the terminology. But anyhow, look at all those regions involved in vision. Okay, so that's the point I'm trying to convey across. How is information from all these distinct regions involved in specific aspects of visual processing, for example, shape, color, motion, depth, integrated into a unified and correct representation of the external object? As discussed earlier in the previous presentation, part one, even if this could be explained, it would be like explaining how a TV converts a cable or aerial signal to an image on the screen. Amazing signal processing, to be sure, and not something a brain has processed like evolution could ever devise, but it doesn't explain the much harder problem of how you, the ghost in the machine or mind, is able to perceive that it's become aware of through the senses, especially uh, see, observe, and think about the image. If no one even understands how it works, despite intensive research into this, saying that it evolved carries no scientific weight. Rather, it is just blind faith in scientism, materialism, evolutionism, and physicalism, atheism, or whatever religion is subscribed to. Even after spending numerous resources, some smart people eventually solved the visual binding problem. Would this be evidence that the ability of the brain to process visual information was achieved without intelligence and planning? Rather, it would be evidence that it took great intelligence to encode the information into the DNA for the brain to do this. Okay, so here's a there's so many unresolved questions and mysteries about the brain. Just ponder about the above statement again by writer James Lafano. The twin unfathomable profundities of nature and nurture. First, how just a few thousand genes might instruct the arrangement of those billions of neurons with their hardwire faculties of language and mathematics. And second, the physical basis of that all-encompassing property of neuroplasticity by which the brain incorporates into itself the experience of a lifetime. So let's have a brief look at language. Now, Broca's area in the left, shown in the diagram there in the left inferior frontal cortex, is usually associated with language production output, that is speaking. In the superior posterior temporal lobe lies Wernicke's area, shown in the diagram, and this is associated with understanding language. Now, running around the lateral sulcus visuosylvius, shown in diagram, in the left hemisphere of the brain, there is a sort of neural loop that is involved both in understanding and producing spoken language. In this loop, Broca's and Wernicke's area are connected by a large bundle of nerve fibers called the acute fasciculus. This language loop is found in the left hemisphere in about 90% of right-handed people and 70% of left-handed people, language being one of the functions that is performed asymmetrically in the brain. Surprisingly, this loop is also found at the same location in deaf persons who use sign language. This loop would therefore not appear to be specific to heard or spoken language, but rather to be more broadly associated with whatever the individual's primary language modality happens to be. The information in the fibers of acute fasciculus travel from Wernicke's to Broca's area. There may be reciprocal connections going the other way, but it's not confirmed. Also, bilingual patients do not necessarily use the same bit of cortex for storing the names of the same objects in two different languages. Okay, but that's not those are not the only language areas. By the way, children have an innate capacity to learn language, something that adults find much more of a struggle. Now, electrical stimulation studies during neurosurgery and more recently functional neuroimaging studies, as indicated in the diagram above, have found that the brain regions involved in language only approximate those indicated by all the textbooks, as exact locations differ unpredictably among individuals. Many of the sites that elicit interference with language in response to stimulation are outside the classical Broca's and Wernicke's area language areas just discussed. Hence, whilst Broca's and Wernicke's area are involved in expression of speech and comprehension of language, respectively, they are not the only areas involved. Modern imaging techniques have confirmed that multiple areas of the brain are involved in language. So in the brain, from this study, it says in the last 20 years, there has been an explosion of research into the neural basis of language processing. This has clearly established that spoken and written language relies on concurrent activation in multiple brain areas. Okay, I'm not going to read the rest of that in the slide, but I'm just showing you here that here there's a lot of areas, even the cerebellum. Okay, so there's a lot of areas involved in language. Now, however, localizing aspects of language function to certain brain regions does not lessen the mystery of how it works, any more than knowing that engines power cars informs you how engines work. Now, we are told in *Sapiens: A Brief History of Humankind*, a book by Yuval Noah Harari, that the appearance of new ways of thinking and communication between 70,000 and 30,000 years ago constitutes a cognitive revolution. As to what caused this, the author admits to not being sure, but states, as I quoted there, I've given the quote up there on the screen, he says, the most commonly believed theory argues that accidental genetic mutations changed the inner workings of the brains of sapiens, enabling them to think in unprecedented ways and to communicate using an altogether new type of language. We might call it the tree of knowledge mutation. Why did it occur in sapiens DNA rather than in that of Neanderthals? It was a matter of pure chance, as far as we can tell. Hence, evolution waved its magic wand and we went from caveman grunts to Shakespeare. Wow. Magic bullet genetic language mutations such as the tree of knowledge mutation being seriously espoused as responsible for a unique way of thinking and communicating are simply not plausible to explain something so complex. To me, it's about as likely as a lightning strike hitting your car and upgrading it from a Volkswagen to a Mercedes. It's never going to happen. Now, think of the waiting time involved in fixing the genetic mutations into the genome to incorporate or integrate all those cortical regions so they can also be used for language processing. How many, I don't know, but there would have to be thousands of such mutations. The problem, in a nutshell, is that calculations show that it would take way too long for these specific mutations to rise and become established. Addressing the waiting time problem in a general way, according to John Sanford et al., who's an expert in genetics, and given the quote on the screen here, they looked at the waiting time problem. It says, we have used comprehensive numerical simulations to show that in populations of modest size, such as a hominin population, there is a serious waiting time problem that can constrain macroevolution. Our studies show that in such a population, there is a significant waiting time problem, even in terms of waiting for a specific point mutation to rise and be fixed minimally about 1.5 million years. We show that the waiting time problem becomes very severe and more than one mutation is required to establish a new function. And go on to say, in a population of 10,000, the establishment of a string of just two specific codependent mutations tends to be extremely problematic, conservatively requiring an average waiting time of at least 84 million years. For nucleotide strings of moderate length, 8 or above, waiting times will typically exceed the estimated age of the universe, even when using highly favorable settings. I'm just going to take a sip of water. Now, different people may have different calculations, but it doesn't change the improbability of it all in any meaningful way. Fixing the genetic mutation into the genome to incorporate or integrate all those cortical regions with language will not happen in 70,000 years, nor not in 6 million years, around a time when we allegedly branched off from a common ancestor with the chimps, not even in the evolutionary ages given for the universe. Okay. Now, as the obvious alternate explanation is that it was designed by a superior intelligence, that is God, I guess people who have ruled out God as creator a priori feel they must propose some explanation, no matter how impossible it may be. Now, just to go elaborate a bit more, according to Cornell, John Sanford, who was a Cornell University professor, he says in his book Genetic Entropy, furthermore, the nature of selection is such that for one nucleotide selecting for one nucleotide reduces our ability to select for other nucleotides, selection interference. For this reason, simultaneous selection for many weakly beneficial mutation is largely ineffective. In the last 6 million years, selection could maximally fix about 1,000 unlinked beneficial mutation, creating less new information than is on a typical page of text. There is no way that such a small amount of information could transform an ape into a human. Now, even if the genome, the DNA difference between chimps and humans were as little as 1%, as used to be widely touted, this still represents around 30 million nucleotide differences, and hence in an evolving hominid line, around 15 million nucleotide changes will be needed to take place, compared to the 1,000 changes at most that could have happened that has become fixed in that time. But the problem for evolution is now compounded because the chimp-human difference is now known to be not 1%, but likely at least 5%. So the hurdle for evolution is even more insurmountable. A five-fold increase in difference now means some 75 million nucleotide changes since the imagined common ancestor. The problem is worse still, as in addition to this, the human genome is deteriorating on a downward spiral towards mutational meltdown. This is due to the accumulation of genetic mutation at the rate of some 70 to 100 point mutations per person per generation. The figures were given by some people vary, so I just put that in there. So the exact figure is basically debatable. With natural selection powerless to stop it. Making matters even worse for evolution theory, the amount of junk DNA believed to be in the genome has shrunk considerably of late. The proportion evolutionists think is functional has now increased from about 3% to 80% or more. In the end, it may well approach 100% functionality, but that is not known yet. A high functionality rate is a problem because it makes it far more likely for any mutation to be harmful, not neutral. As stated by creationist Eric P., most harmful mutations have only very mild effects. That's good in the short term, but that also means natural selection usually can't filter them out. And it goes on to say, even many ardent anti-creationists recognize this problem of having too many mutations, Biochemist and textbook author Larry Moran states, if the deleterious mutation rate, this is the harmful ones, is too high, the species will go extinct. It should be no more than 1 or 2 deleterious mutations per generation. Eric states that if mutations drive species to extinction, they couldn't have been around for millions of years. And as it's an extremely important issue, the waiting time, in determining whether evolution is possible, I'll just discuss another example. So just a brief detour, and then we get back to the brain. Evolutionists Dennis Bramble and Daniel Lieberman counted about 16 derived features of the human skeleton with functions adapted for running that were first present in a group of Homo erectus, 15 of them, and Homo sapiens, one additional derived feature, but were not present in the Australopithecines. The features are said to be necessary for head and body stabilization to allow counter rotation of the trunk versus head and hips, trunk stabilization, stride length, thermoregulation, and stress reduction. Now, Anne Gorger, an expert in development biology, looked at how long it would take to transform an Australopithecine such as Lucy so that it runs like a human. Okay. Now, she did calculations by evolution. She investigated calculations by evolutionary experts. And apparently, the waiting time for one mutation to occur and become permanently present that is fixed in a primate lineage is about 6 million years. And if 2 mutations are required, if the first one was neutral in its effect, would take 216 million years. This is just for 2 mutations. To generate the anatomical changes necessary to make an Australopithecine walk and run like a human by evolving all 16 anatomical features would require at least 16 mutations. Likely more if each anatomical feature required multiple mutations. According to Gorger, many of the anatomical changes seen in Homo erectus had to occur together to be of benefit. Individually, they'd be useless or even harmful. And she says, and getting all 16 mutations to appear and then become fixed within 6 million years since our alleged split with chimps, let alone the 1.5 million that it apparently took, that is for the Australopithecines to evolve into Homo erectus, can't have happened through an unguided process. Now, 216 million years is just for 2 mutations. To generate the anatomical changes necessary to make an Australopithecine walk and run like a human would require, according to Anne Gorger, dozens, if not hundreds or thousands, if it could happen by random mutation of all. For all these mutations to appear and then become fixed within the 1.5 million years or so, it apparently took the Australopithecines to evolve into Homo erectus, or even within the 6 million years since our alleged split with the chimps, can't have happened through an unguided process. According to Anne Gorger, in looking at the odds of it happening, she says it strains credibility to think that all 16 anatomical features evolved for maturity in that same time frame, especially if each required multiple mutation. Given these numbers, it is extremely improbable, if not absolutely impossible, for us to have evolved from hominin ancestors by a gradual, unguided process. All right, so now we'll get a bit back to the brain again. Okay, so I just wanted to show this waiting time problem that sometimes I refer to, that it's a very serious problem. Anyhow, now, as an example of the intricate wiring of the brain circuits, consider the release of the hormone melatonin from the tiny pineal gland, the main site of its synthesis in the brain. Now, melatonin has various physiological roles, including circadian rhythm control, that is the internal body clock that tells our bodies when to sleep, rise, eat, etc. The hypothalamus of the brain, which controls both the autonomic and the endocrine or hormonal system, is involved in regulating the body's circadian rhythm through its circuitous connections with the suprachiasmatic nucleus, SCN, through its connection of this nucleus that this nucleus makes with the pineal gland. All right, so Let's sort of block a bit of a diagram up there. Now, the tiny suprachiasmatic nucleus, SCN, which is situated near the optic chiasm, that's where the optic nerves cross over. It receives direct input from the retina's ganglion nerve cells via the retinohypothalamic tract, thereby allowing eye-like exposure to synchronize the body's 24-hour internal clock. Now, the suprachiasmatic nucleus controls circadian rhythm through connection with another hypothalamic nucleus, the paraventricular nucleus, PVN. The PVN, in turn, signals the sympathetic preganglionic nerve cells located in the thoracic spinal cord. These sympathetic nerve cells then synapse on postganglionic nerve cells in the superior cervical ganglion. From there, the neurogenic postganglionic nerve fibers finally innervate the pineal gland. When I say neuroadrenergic postganglionic nerve fibers, I mean they emit noradrenaline as their neurotransmitter. Noradrenaline, also known as norepinephrine. Now, the pineal gland produces melatonin in darkness, but not in bright light, and so the amount of melatonin secreted is regulated by the incoming nerve stimulus, which in turn is determined by the amount of light stimulated signal received by the suprachiasmatic nucleus. The melatonin is released into the blood, and the level of hormone in the brain influences the 24-hour day-night rhythm. It readily passes the blood-brain barrier and accumulates in the central nervous system at substantially higher levels than exist in the blood. Okay. Now, just checking. Are you still there, Donny? You there,
Peter?
Donny?
Yeah, Peter, we're good. We're good,
just
brother.
checking, so I'm actually sure that we haven't been cut off. Okay, fine.
No, it's going smoothly. You're doing great.
Okay. All right. Now, so this description I went through, it admits all the complex interactions between neurotransmitters and receptors at the synapses. That is the connection between nerves, as well as the manufacture of the hormone and how the receptors of melatonin at the target cells got there. Now, melatonin receptors are found throughout the body in places such as the brain, the retina of the eye, the cardiovascular system, the liver and gallbladder, the colon, the skin, the kidneys, and many others. The actions of melatonin are mediated by two G protein coupled membrane receptors, MT1 and MT2. I'll discuss in a few minutes more about G protein coupled membrane receptors, which are quite actually a complex cascade of events. Now, melatonin feeds back to the suprachiasmatic nucleus via melatonin receptors located in the suprachiasmatic nucleus, allowing the suprachiasmatic nucleus to decrease neural firing if melatonin levels are too high. So there's a feedback system as well. Now, these MT1 and MT2 melatonin receptors have a general structure designed consisting of 7 transmembrane spanning segments. I'll show you what I mean by transmembrane in a short bit. Now, and also these receptors have, I mean, they're about 350, 360 odd amino acids long. Okay, these receptors. Now, if this all seems very complicated, then ask yourself, how could such an intricate pathway or setup be put together except by purposeful design? Is it feasible that natural selection, sorting, random mutation could put the information into the DNA to construct such a pathway, given the steps and complexity involved? What would be the waiting time involved of getting such information fixated into the genome? All right, so just a bit about chemical synaptic transmission. Now, chemical synaptic transmission is the predominant form of synaptic communication in the central and peripheral nervous system, and it's mediated by substances called neurotransmitters. Now, each neurotransmitter produces its effect, whether postsynaptic or presynaptic, by interacting with a receptor molecule in the cell membrane. Now, there's 2 superfamilies of receptors that specifically modulate or participate in chemical synaptic neurotransmission. The ligand-gated channels, which are often referred to as ionotropic receptors, and they kind of mediate direct or fast neurotransmission. And there's, as I alluded to before, G protein coupled receptors, often referred to as metabotropic receptors, and are said to mediate indirect or slow neurotransmission. Okay. Now, here is the structure of a ionotropic receptor, so it mediates fast or direct neurotransmission. And these receptors are a world of complexity in themselves. The direct or fast effect involved on membrane potential at synapses are as mentioned by neurotransmitters known as ionotropic receptors, and basically these open or close various membrane channels. Okay, that's all they do: open and close the channel. And one of them, such receptors in the nervous system, is a nicotinic acetylcholine receptor, which are composed of 5 subunits. It's called nicotinic because nicotine can also bind to these receptors as well. So it's kind of given the name nicotine. Now I'm just sort of one of my mouse cursor there. Now the subunits are arranged. Now this consists of 5 subunits here. You can see them here. 1, 2, 3, 4, 5. Here you can see the front one subunit here, how they're arranged. This is a cell membrane here. This is inside the cell. This is outside the cell, extracellular domain here. This is the ligand binding domain. So this is a subunit 1, 2, 3. Most of the subunits are in the extracellular domain. Here's the transmembrane spanning segment. So when I mentioned before that the melatonin receptor has 7, these ones have 4, but these actually subunits are actually longer. They're about 4 up to 500 amino acids in length. So even that, how long does it take for that? Now, some of the subunits are identical. So of these 5, it depends on which version of the receptors you get, or some may be different. But even if you have one of them, what's the waiting time to get 4 to 500 amino acids in the correct order? And these things also operate. It's like a lock and key thing. What happens is that you have these transmembrane spanning segments that they go sort of back and forth across the membrane. This is a large extracellular domain, a large sort of thing sticking out here between M3 and M4. But if you look down from the superior, looking down upon a cross section of the receptor, you can see how these membrane spanning segments are arranged. You see the M2 is around the central pore here, which is where the neurotransmitters pass, and you have the M2. And what these M2 spanning segments, they believe when they're closed, they're kind of kinked, sort of kinked inwardly, and it's kind of lined with a believe sort of amino acids. I think it's leucine or something like that, which basically is hydrophobic, meaning water hating. And so basically that it closes off when these line up. It closes the channel. Now, when a neurotransmitter binds here, there's 2 sites that have to be bound to. Acetylcholine is the neurotransmitter. Somehow this does a conformational change in these subunits, which moves these hydrophobic amino acids out of the way and replaces them hydrophilic, water-loving, and opens it up so you can have these cations, positive ions, kind of moving through sodium. So in a normal state, sodium would kind of go inside, potassium would go outside. Okay. Now so yeah, and so this is a sort of getting my cursor back there. So the thing here is what I hate this thing of the cursor. With a lock and key approach, it's not just getting the subunits arranged, but think of it, you have to have these subunits have to be arranged so that they fit with the neurotransmitter. There has to be the key has to, otherwise they're useless unless they unless the neurotransmitter, in this case acetylcholine, can bind to the receptor and basically move these cause a conformational change, which open up the channel. So again, this is a quite elaborate, sophisticated system, and not something that is likely to happen by chance anytime soon. But this next one, and I apologize for my poor hand drawing diagram. That's the only one in this slide thing. But the next one is the whole thing is more complicated. Maybe not the receptor, but the cascade of events is more complicated. So this is a G protein coupled receptors on the right compared to, say, the direct transmission, which is shown on the left. So they're basically as with the direct transmission, there's a lot of different versions of the channels, and with the G protein coupled receptors, there's a lot of different versions which do different things. So it's not just one; there's lots of difference of them. So how these things work is basically if I can get my cursor in here. Hang on. Yeah, here we are. So you have a first messenger, which is the transmitter or neurotransmitter. I've just labeled it transmitter. That binds to basically the receptor. So it may be something like serotonin. Now the receptor is coupled to a G protein here, a G protein complex, which is an enzyme complex. So after the first messenger, basically the transmitter binds to the receptor, this complex activates an enzyme basically called, say, in this instance, adenyl cyclase here. You can see it activates it here. And this converts ATP. The ATP is the energy currency of the cell, and that converts it into this ATP into the second messenger, cyclic AMP. Now, so cyclic AMP is a second messenger, and that may open membrane channel for example, by activating a protein kinase enzyme here, which in turn phosphorylates, that is, as a phosphate group, to a substrate protein here. And now this substrate protein could be the channel or itself, or a protein that acts on a channel and which causes, say, this causes the potassium channel to close. Now, so there's a lot of these receptors in the brain with many different versions. I don't know how many there's 100 trillion or more connections likely in the brain. And if these are the primary form of transmission, then, who knows? There's just a lot. If you count it, someone again calculated, if you have 100 trillion connections in the brain and you counted each one a second, it'd take you 30 million years. So, yeah, I don't. Anyhow, so that now, now let's go on here. This book actually was why us, how science rediscovered the mystery ourselves. Had a lot of interesting information in it. It was a while ago I read it, but he had a lot of interesting quotes, so that's why I quote him, which is sort of apt to this presentation. So there's another quote from James Levano. He says, "Science may have a thorough understanding of the workings of the heart and lungs, but the unpreprocessing 3 pounds of brain tissue confined within our skulls, like a vast intellectual black hole, absorbs the most searching forms of scientific investigation. Its most elementary currency of a single thought defies any materialist explanation, let alone how that thought might be rational rather than irrational, inspired rather than banal." Now, this leads into a brief discussion of the mind, which we'll kind of look at next. Now, the mind, which I regard as distinct from the brain, allows you to reflect vividly on the past, to make informed judgments about the present, and to plan rational courses of action in the future. It endows you with a seemingly effortless ability to form pictures in your mind, to enjoy music, to dream, to dance, to fall in love, cry, and laugh. You think of your mind, but the mind and the thoughts that it produces are not physical. According to the Neuroscientist book, much is left to discover about the truth of free will, and plenty of what we know, plenty of what we do know is open to debate. Nevertheless, neuroscientists now agree that when we make voluntary actions, it's not because of any ghost in the machine. It's not because there's any sort of mind which is independent of the brain. Instead, there are brain processes which are accompanied. Just checking the heater isn't working too well. And it's not because there's any sort of mind which is integrated, which is independent of the brain. Instead, there are brain processes which are just accompanied by a conscious experience, just as visual perception is a product of activity in the visual area of the brain. These specific brain processes give us a sense of control, a sense of being in charge of our body and our lives. Now, the materialists use elephant hurling tactics. For example, neuroscientists now agree that when we make voluntary actions, it's not because of any ghost in the machine. It's not because there's any sort of mind which is independent of the brain. So they use these elephant hurling tactics to push their view that there's no immaterial mind or ghost in the machine, that what we think is that mind is just an experience that accompanies brain activity. In other words, you are your brain. However, the reality is that neuroscientists are not in a position to make such a declaration, except as a statement of philosophical belief, as they don't have the data to support such an assertion. In other words, they don't know what the mind is, so how can they know what they don't know? Now, the mind, as stated by neurologist Richard M. Riestack, mind is not a physical structure like the brain. It is not a thing. Mind has no visible form, no aroma, no taste. It can't be held in the hand like the brain. Thoughts, the products of the mind, do not require physicality to exist. Thoughts, however, are meaningless without minds that can interpret them. Now, neuroscientist Joseph Green writes, indeed, the lack of neuroscientific theories explaining the functioning and features of neural circuits represents a fundamental deficiency in our understanding of the brain and mind that is easily identified. Given recent technical breakthroughs, it is reasonable to believe that the scientific community now understands the mental activity captured by cutting-edge technology, yet this is incorrect. Our theoretical understanding of the brain, unlike that of other physical systems, remains extremely limited. He also says, in reality, even in the most optimistic case, neuroscience may be decades away from being able to inform critical ideas in philosophy of mind. In other words, philosophical theories addressing the mind-body problem are, for the most part, unlikely to be validated or invalidated by neuroscientific findings in the upcoming years. He also says, at present, scientific theories do not constrain most philosophical ones. This implies that the current dogma that pervades neuroscientists, materialist monism that can be simply stated as we are nothing but our brains, is established by intellectual pressure rather than solid scientific evidence. Okay, now here, let's try to get my cursor back. Now, the study indicated above investigated the organization of resting state networks in high-functioning adults who had childhood hemispherectomy, basically removing nearly one hemisphere of the brain. So that's most part of the brain, although they kind of leave the brainstem intact and usually the diencephalon intact as well. Although in some cases they may take parts of the diencephalon, but anyhow, in some, and the authors of the study say in some, the current study provides evidence on the neural reorganization that produces compensated cognition after the surgical removal of one hemisphere. Functional connectivity of the human brain as measured with resting state functional MRI leaves open exciting future questions for task-based functional localization and hemispherectomy. Insights from these rare patients argue that intrinsic mechanism of brain organization in only half of the typical available cortex can be sufficient to support extensive cognitive compensation. The research, and I'm going to say, And commenting on this study by Whitney Clavin, he states that the research team performed magnetic resonance imaging MRI scans on patients, all of whom received the surgeries as children and now have relatively normal, and again to say relatively normal cognitive abilities. The patient scans were compared to those of healthy individuals. The results show that brain networks in hemispherectomy patients, networks that control waking, talking, and other functions, were remarkably intact. Despite missing an entire hemisphere, we found all the same major brain networks that you find in healthy brains of two hemispheres, said Dorrit Kleeman, lead author of the new report. Says Kleeman, it's truly amazing what these patients can do. Yes, they have challenges, but their cognitive abilities are still remarkably high functioning given that they are missing half of the brain tissue. We need to understand how this is possible with only a single brain hemisphere. An important question about plasticity, reorganization, and compensation. Okay, in this case, a 7-year-old female with medically refractory epilepsy secondary to Rasmussen encephalitis was treated with a complete right-sided anatomical hemispherectomy. At the time of follow-up MRI, 11 years post-operatively, she was seizure-free and able to walk independently with mild spasticity of the leg, sort of stiffness or whatever. Now, she was of normal intellect and applying to higher education. I won't read through the whole thing. Now, the present case is testament to the extraordinary plasticity of the young human brain. Here, left-sided motor control relocated from the diseased, excised right hemisphere to an anatomically comparable area in the left hemisphere. Interesting, this functionality did not relocate to a random cortical area, but to an area that already contained the prerequisite cellular architecture and white matter connectivity required to control movement. The present patient demonstrates a remarkable neurological recovery after hemispherectomy. The use of functional MRI illustrates the functional reorganization of the motor cortex to the contralesional hemisphere. It is supportive of existing literature highlighting the potential for transposition of the motor cortex to the remaining hemisphere. All right. Now, hemispherectomy, I brought this up because it highlights 3 issues. Firstly, ever wondered how the supposed evolutionary process, one that has no aim, plan, or intelligence, could create the ability of the brain to reconfigure itself when half of it is removed? For example, as mentioned in the previous slide, the extraordinary plasticity of the young human brain, where left-sided motor control relocated from the diseased, surgically removed right hemisphere to an anatomically comparable area within the left hemisphere. If the brain was a product of evolution, how is it even possible for someone to survive the removal of nearly half the brain, that is one cerebral hemisphere, let alone for the brain to reorganize itself in such a way? There would not have been any half-brain hominids, that is ape man, in the alleged environment of evolutionary adaptiveness, that is, for example, the African savanna. So a half-brain person is novel, unless one is proposing that the magic wand of evolution plans intelligently for such contingencies, then the answer it would seem impossible for the brain to reorganize and adapt in this extraordinary way if it was created by an evolutionary process, one in which only the fittest survive. Now, the second issue, also, if you can have a normal intellect with nearly half your brain missing, about the same size in terms of brain mass as a small Homo erectus brain, why, as alleged by evolutionists, would brain size increase in supposed hominins be driven by intelligence in the alleged environment of evolutionary adaptiveness, as what would be the benefit of getting a bigger brain, particularly given the costs? It's not like they was designing space rockets in that supposed environment. Now, the above question is from, the above quote is from Ian Tattersall, who was curator of the Spitzer Hall of Human Origins at the American Museum of Natural History. I'm not sure what he does now. Now, it says brain tissue is metabolically expensive, so there must have been some very considerable advantage to larger brains. Presumably, the dividend was received in terms of greater intelligence, although precisely what this might have meant in terms of abilities or context remains unclear. Note that the presumed dividend, let's call it benefit, was greater intelligence, but intelligence or abilities in what remains unclear. There would be no direct benefit, that is, dividend in terms of greater intelligence in slightly increasing brain size over millions of years or hundreds of thousands of years, when, for example, a small Homo erectus brain was large enough. Without any significant selective value, large brain size could not evolve even if evolution was possible, which it is not, particularly considering the high metabolic energy costs of big brains. There would be a cost in terms of using extra energy for no gain, an evolutionary no-no. So brain tissue is metabolically expensive. The human brain accounts for about 2% of total body weight, yet consumes about 20% of the energy which is used by the human body, or at least at rest, the basal metabolic rate. Now, as the iconic and false depiction of evolution, the march of progress of alleged ape men, I think that was discussed in a presentation by Carl Werner a little while ago in another presentation, has played a role in indoctrinating the public in evolution. So has the supposed incremental march of progress in brain size of alleged ape men also done similarly, that is, indoctrinate people into believing evolution must have happened. Colin Barris, a freelance science writer, did a recent story on the subject in Neuroscientist. He quotes paleoanthropologist Jeremy De Silva saying, "Why would selection favor bigger brains where small brain humans can survive, when small brain humans can survive on the landscape?" I'm just going to take a sip of water. Note that they're not asking a question because of hemispherectomy data that I presented before, but rather because of evidence of small brain fossil specimens as determined by cranial capacity that I believe are exhibiting evidence from artifacts of enough intelligence to survive in their environment. According to Barras, an answer to this puzzle is beginning to emerge. It looks like brain expansion began as an evolutionary accident and then led to changes that caused this growth to spiral. Surprisingly, the sorts of changes that drove this expansion could also explain a more recent 10% reduction in human brain size. What's more, this suggests that our brain size may shrink further still and may even cause humanity's demise. Oh dear. Anyhow, Colin Barris also writes, but Baudet has an idea. Emily Baudet is a paleoanthropologist at the University of Cambridge. So Barris says, Baudet has an idea. Let's assume a meat-rich diet led to an energy surplus, and some of that energy fueled brain growth simply because the extra energy had to go somewhere. But as brains expanded, they ran into a problem. At some point, just because the brain is getting bigger, you have no space in the brain case, says Baudet. The surface of the brain, which was already folded in small brain humans, would have gained an even more elaborate pattern of folds and furrows as it pressed against the skull. This folding potentially brought neurons from distinct brain regions into closer physical proximity, she says, allowing for greater connectivity between those regions. Purely by chance, some of those connections might have made it easier for humans to talk and make tools. In other words, brain expansion may initially have had no survival advantage, but then serendipitously acquired one, at which point natural selection would have kicked in. Nice, just a story, but that's all it is, a fictitious story. Now, the third issue is, which particularly relates to the mind, is materialism predicts that the mind can be divided into parts. Hence, removing nearly half the brain, that is nearly half the parts, should produce a fractured, split mind according to materialism, but the mind remains intact. So, a professor in neurological surgery and pediatrics, Michael Egnor, writes, materialism predicts that the mind, like the brain from which it arises, is metaphysically complex, in the sense that it is divisible into parts, just as any material thing has dimension and physical relation, and as such can be split in one sense or another. And he says, hemispherectomy, which is a neurosurgical operation distinct from but related to commiserotomy, This demonstrates retention of unity of mind despite complete removal of entire hemisphere of the brain to stop intractable seizures. Now, in dealing with metaphysical questions at hand, Egno considered relative neuroscience studies and experiments, including split brain research, commiserectomy, hemispherectomy, the new phrenology, that is correlating brain states with mind states, cortical stimulation, epileptic seizures, craniopagus twins, which are twins conjoined with conjoined twins fused at the cranium, functional MRI studies of patient and persistent vegetative state, near-death experience, and Benjamin Libet's free will experiments using the redness potential. So, example, after a hemispherectomy, he says, removal of an entire hemisphere, Engel states, there is no evidence of splitting of consciousness, meaning intellect, will, and unitary sense of self. That's you removed up of an entire hemisphere. There is no evidence of splitting of the consciousness, meaning intellect, will, and unitary of self. From his evaluation review, Michael Egnor concludes that perhaps the most apt formulation is that for sensation, perception, motor movement, imagination, memory, and emotion, specific brain states are necessary and sufficient, while for intellect and will, brain states are ordinarily necessary but not sufficient. Intellect and will, the characteristic powers of the rational soul and Aristotelian psychology, are not powers caused by matter. The most parsimonious explanation for the perceptual intellectual dichotomy is that intellect, in contrast to perception, does not arise from brain states, but requires a source other than matter. Okay. All right, so we're getting towards the end. So the next thing is to briefly just discuss near-death experiences. Okay. Now, whatever you think of near-death experiences, if they are real, then they indicate the mind is immaterial, that is separate from the body and survives death. Now, the case for near-death experiences, that is the existence of at least some initial credible indications of afterlife consciousness, is given by philosopher and theologian Gary Habermas. In his chapter in the book on given there, I think it's the Minding the Brain. In his chapter, Habermas presents evidence for the reality of near-death experiences using plenty of examples with corroborative accounts that are difficult to explain in any other way than that the near-death experiences happen as claimed. According to Habermas, most near-death experience critiques and alternative hypotheses tend not to focus on the outside corroborative part, instead postulate conditions that are internal to the individual near-death experiencer, such as oxygen deprivation, temporal lobe seizure, drugs, exaggerations, false statements, hallucinations, or either a dying or awaking brain. That is, they rely chiefly upon the interior physiological conditions and of psychological states of the near-death experiencer's mind or body. Now, limiting himself to the cases referred to in his essay alone, there are over 300 evidential near-death experiences cases, that is, where external circumstances argue for their veracity. Hence, Habermas argues that almost every one of them must be mistaken for the natural supposition against near-death experiences cases to work. He states, most evidential near-death experience cases seem to occur during a state of cardiac arrest due to ventricular fibrillation. It is exceptionally well documented that during such a condition, the heart stops and at least higher cortical brain activity usually ceases in a matter of just 10 to 15 seconds or so afterwards. Now, I've seen this happen and many, many years ago, and believe me that when the person actually arrived, believe me, when it happens, that person is not thinking. Believe me, this person is not in a state to observe anything going on in a room. Now, mathematician and philosopher William Dembski discusses informational realism and touches on the near-death experiences. He says the ontology is responsible for the existence of a mind-brain problem that is materialism, as those artificially restricting the creation and flow of information. He gives an example of this restriction of information flow accounts given by people having had a near-death experience. You can tell what was talked about in other rooms during the near-death experiences. With materialism, this is impossible. If you're essentially dead, you can't know what's going on in your room, let alone another room. And so such information must be through some subsequent communication of the information via ordinary channels. Now, Debsky points out that materialism makes demands on what types of information certain types of information sources can generate. For example, that the mind must be reduced to mechanisms, as materialism sees all things as composed of body parts that operate by mechanical principles. Now, in regards to near-death experiences, if a person can look down upon their body from the ceiling and observe things going on in a room or other rooms or places, then that is impossible from a materialist perspective if the person is essentially dead. It indicates an immaterial mind that is separate from body and also a mind that survives death. All right, so now another one or two quotes from James Fanu, and then I'll be finished. He says, finally, the big science of neuroscience, observing the brain in action, has revealed processes that defy all imagining. How every detailed nuance of the three-dimensional world is generated from within the dark recesses of our skulls, deconstructed and reconstructed within a fraction of a second. Or how the brain categorizes our memories into different baskets, shifts them from one to the other, and somehow maintains them as permanent record in those ever-changing neural circuits, or how, contrary to every known law of nature, non-material thoughts and emotions directly influence the physical structure of the brain. He also states, by contrast, it now seems deeply irrational for materialist science to deny the exceptionality of the human mind and to insist that the sense of self and free will are no more than illusions generated by the workings of the brain. It is certainly irrational to assert the truth of the evolutionary doctrine in the face of all the scientific evidence that will contradict it. By evolutionary doctrine, he in the quote said, the mystery of existence is a mystery no longer because of Darwin solved it. So that's just complete garbage. So I shouldn't use that so blunt language, but that's what I think anyhow. Okay, so this is the last slide now. Okay, no one knows how the mind works and how it links with the physical activity of the brain. Claims that the mind and brain are products of evolution, that they are one, and that you are your brain, are simply Darwinian pledges of allegiance. The materialist or reductionist insists that everything can be reduced to matter and its movements, which in the brain means mental events such as thinking, emotion, and awareness must break down to interaction between nerve cells and associated molecules if mind and consciousness are physically part of the brain. As the mind cannot be described in reductionist terms or reduced to matter, it ironically falsifies the very philosophy it is used by some to espouse. To me, it is inconceivable that the human brain, along with the non-physical mind, could have created itself by a process devoid of intelligence, plan, or purpose. It would be impossible. The human brain, including mind, does not give up its secret easily. There are so many mysteries. The thing is, if it's so horrendously functionally complex and so much is beyond our understanding, how on earth can evolutionists insist that there is no doubt that the brain and mind evolved? In effect, they are saying that something as brainless as pond scum created, although they don't know how, except for just so stories. In fact, one hears all kinds of just so stories about how the human brain evolved. All seem to be acceptable to some degree in academia, yet mention the only explanation that makes sense, that is by intelligent sign of a creator God, and somehow that is unacceptable. There is something deeply wrong with such logic. If the issue was about science, both theories would be given time and fair treatment in academia and the media, etc. That it is not indicates the issue is not about science, but about different worldviews. At its core, it's a spiritual issue. The evidence for design in the human brain is self-evident, but the same cannot be said for evolution. From the starting position of pond scum, a single-cell algae or something, could the human brain, containing the most complex arrangement of matter in the known universe, doing feats no man-made machine can even come close to, have created itself by an evolutionary process without intelligence, sign, plan, aim, or purpose. In my opinion, it's about as likely as me being able to jump to the moon without rockets. Romans 1:22 says, professing to be wise, they became fools. Perhaps that is what is going on. And that is the end of my presentation, Donny. You there, Donny?
Yes, Peter, I was backstage putting you front and center because that presentation was incredible. I know I will be having to rewatch part 1 and 2. Extremely comprehensive, Peter. I appreciate all the work, time, energy, and effort you put into this 2-part presentation. Very technical and also important. Just a real epic dismantling of human evolution. In my opinion,
I'm
this.
just moving my heater closer to me because the heater didn't work. Sorry,
Oh,
continue. Thank you.
no worries, no
Just
worries. In my opinion, what you presented over the course of these 2 videos, Peter, is what's called preclusionary evidence. And what that is a way, I think, to discriminate or differentiate between the models, creation and evolution, because the evolutionists simply can't explain this data. They simply can't explain this evidence from a naturalistic or evolutionary starting point, whether they want natural selection, mutations, epigenetics. And so, because as we know in the creation evolution debate, brother, there's a lot of overlap. There's a lot of what's called agnostic lines of evidence, right? Where both models can explain the data. But in this instance, in this 2-part presentation, only the creation model can account for this. Earlier in your presentation, you touched on so much, but you made one point. You had one slide talking about how orderly, parallel nerve fibers, these interconnected, highly sophisticated systems, what you'd expect if evolution was true, correct me if I'm wrong, would be a mess, something more akin to spaghetti. But we see something that screams design. Is that right, Peter?
Because it only works with what it got at the time, and it can't. And once it would establish those connections, it's very hard to go back and reverse it and tie it in with other stuff. So it's going to be more. It's going to be a dog's breakfast arrangement, not something that looks like it's been planned ahead of time and then put in like an engineer would do, creating an integrated circuit board. Would it can't go back and make the corrections later like that. It's be very difficult because the complexity, as I hope I've indicated, there's so many variables in the brain, whether it's hormones, whether it's neurotransmitters, whether it's receptors, and all these different regions involved in, say, in language process, the vision. You got to it's so you just can't go back and basically if you want to add something then redo the other, because then you have the waiting time problem to do that to go back again. So it's just I don't know how you can you'd have to say that the evolution has a mind, but then that's not materialism, is it?
Well, you nailed it there, because this is evidence of forward thinking, which points us back to a forward thinker. Natural selection doesn't see the long term. It's not worried about you getting cancer 50 years from now. It's worried about today. It's worried about tomorrow. But what we see in terms of this design, what we see in terms of the epigenome is forward thinking, which means this is forward planning, Peter. And so truly fascinating. I love how you wrap things up more or less with the evidence from near-death experiences. You went into some of Gary Habermas's work. And when somebody truly looks into that, Peter, the evidence is undeniable. I've read stories. I've heard Gary Habermas himself point out. I've recently read a book called Imagine Heaven, where you have these stories of people who die for a time, and they're not dreaming or anything like that, because they leave their body. They can see their body, but then they move over to the next room. And after the fact, they're able to explain in detail the specifics of what was going on in the next room. And then
even if you
confirmed.
weren't dead, if you can leave your body and look down upon it, that still indicates your mind is immaterial and separate from the body. So it's still the materialism. So because in materialist view, you have to have your eyes have. You can only see things through your eyes. Okay, so you can't look down and point yourself from the ceiling if your eyes are in your body. If you ask, so
Right.
yeah,
I find militant critics of biblical creation, which is what we believe in, they like to spend a lot of time focusing on topics and data points that they think they have something good on. They think they have like the fossil record, right? The
yeah.
fossils are fickle, they can tell any story you want. But when it comes to evidence like this,
Mm,
they kind of have to run from it. I don't see them addressing
mm,
it. They don't
mm,
really have any strong answers to these
yeah.
challenges, Peter. Only the
I
creation
I
model can
think the
account for this.
you've heard. I think I looked. I think Micah Beer had a reducible complexity. Dembski had specified complexity. I think I look at this as the kind of word for it. It's overwhelming complexity, but overwhelming
Right.
functional complexity. It's so overwhelming you can't you just it just can't be created by a non-mind. There has to be a mind behind it. It's
Amen.
just I mean it's you basically you're kidding yourself if you think this can be created without intelligence of mind. I mean just from a practical point of view. I mean no no if it didn't have spiritual consequences, then most scientists would say this has a mind behind it. It's because it's that's why to me it's not a science issue. If it was a science issue, no one would have a problem discussing design in academia.
Spiritual
It's
issue.
not a science issue; it's a spiritual issue. That to me, that's the logical because it's so well, it's so strong. The brain evidence for design. You don't lot the complex process. You don't know how a lot of these processes work, but you don't even know what the mind is, and so how just that's what I say. To say mind and brain evolved is just a pled illusion of Darwinism, and well, boy, I can think of a better person to pledge allegiance to than Darwinism. He's not going to reward you in the afterlife,
Amen.
but Jesus
Amen.
Christ will. So I would pledge my allegiance to him instead. That's a better option.
Well, again, that's an excellent point, Peter. I fully agree. If it were not for the spiritual consequences of this evidence, of this incredible design, these highly sophisticated systems, including the human brain, which you presented on, then it wouldn't be debatable. But because there's spiritual consequences, people now
Mm,
have to deny it.
mm.
So,
Yeah.
Peter, I could talk to you about this stuff all day,
Yeah.
but we do have some fantastic questions here. So let's get some of
Yeah.
these up and give you the
I don't know if I can
opportunity
answer
to
them. As I
interact
said,
with them.
the brain has a lot of mystery, so I'll
Yes,
try.
it does.
But if I don't know, I'll say I don't know.
That definitely baffle the evolutionists, that's for sure. It just shows how infinitely complex the infinite God. If I could fit the infinite God in my 3-pound brain, he wouldn't be a God worth worshiping. So we can look at the
Yeah.
human brain and we're just
Yeah.
fascinated by the
Yeah.
design, brother.
Yeah.
Okay, so here we go. This comes in from 21st Century Mind, $10 super chat. Appreciate the support for this evening's program. Can you
Mm.
describe how the physical brain is changed and involved in spiritual/religious experiences? For example, expounding on the research from psychologist William James and neuroscientist Andrew Newberg. I'm not personally familiar with those two scientists, but Peters, is there any thoughts on this?
Yeah. I mean, there's thousands of neuroscientists and psychologists. So yeah, I don't familiar either. I don't know whether they're talking about the sort of God spot experiences. I think years ago I used to laugh about it. It's sort of like is it, my thing was there's probably a Ferrari spot in the brain too, if you like Ferraris that light up. So the thing is, you have an experience, something lights up in the brain, but doesn't, but that doesn't mean that it's it's a spirit. That's religion can be brought down to a spot in the brain. It, it yeah, it's just sort of eh, brain is processing some information. So I don't really, you know, I don't really lay much weight into sort of, you know, of course we use our mind to worship God, and things like that, and and that involves the brain, and so there may be areas that light up, and as we worship God, we may have certain emotions or think certain things or concentrate on things which lights up certain areas of brain, but I don't believe there's a particular God spot or anything like that. But to me, that yeah, it's a bit silly, to be honest. Because because I've done so much stuff on these activation studies using EEGs, well, a certain type of EEG, that where you looked at brain mapping and things light up here, there, and center, and one study shows something and another one shows something else, and so I don't really put too much grain on this stuff, to be honest. Yeah, I don't know. I don't know if that
Is
probably doesn't answer the question, but
no, I appreciate that. Peter, is one of the reasons we can connect. You're talking about these spots that are lighting up, and it is one of the reasons we as humans can connect with God and the supernatural, and animals can't, is because we are body, soul, spirit, and so that part of us, that spirit, especially when we're born again, regenerated, that part of us allows us to connect with God and the supernatural, where the animal kingdom is just body, soul.
yeah. Yeah, well, I don't know how it all works, but but yeah, the Bible says we body, soul, and spirit. I think the soul is sort of, I think it's, I'm not, I'm not an expert on that. I don't know who actually knows, but the soul is more sort of, I guess, related more to the mind, the material mind. Some equate that. The spirit, I'm not sure, but it seems like that's something special given to God, to humans,
Right.
and we created in God's image, and so we interact with God. But we but I think when we do, yeah, I don't know how you, I don't want to try and define something like that, because I just don't know, how you, but we, God is spirit, so we, how you interact, we worship God in spirit and truth, and if something lights up, and when you're thinking all the time, the brain is dynamic, so even if we worship in God, something is going to light up in the brain. You're not going to be brain dead. So something will light up, but it doesn't mean that that spot is for communicating with God.
Right.
It just means that you're thinking about something, and and stuff like that. How precisely what that involves, because if you're communicating with God, it's probably more like a spiritual connection rather than a physical connection. But because we live in this body, we yeah, you have a mind brain interaction, but I don't. Yeah, it's sort of, I don't want to say how it works. I don't know how it works. So probably best. That's probably my best answer, I think.
Very good. No, Peter, I appreciate it. You're doing great. So we got a follow up here. 21st Century Mind, $5 super chat. So lots of support for these programs on the human brain, Peter. So he's wondering. He says a group called Full Preterists deny biology / physics. For anyone in the audience who don't know, a full preterist is basically somebody who claims Christianity, but they believe everything in the Bible occurred in the past, specifically in 70 AD. They would argue that we're currently in the new heaven and new earth. Clearly not true for many reasons. But anyways,
Because,
that's what
yeah, well,
that's what a full
I don't know
preterist.
about that.
Yeah.
But it
I don't
doesn't
know how
seem to
anybody
be.
could be a
It
full
doesn't seem to
preterist
equate in
seeing
my
where the
world,
world's
though. Yeah,
going. So he's
yeah.
wondering. So this group denies biology and physics involved in spiritual events and religious experiences. Would you agree or disagree?
Well, I think I just said, I don't know how we pray to God and things like that. When we pray to God, we use our mind, and when you use your mind, like if you raise your hands and worship God, that means you activate your motor cortex, because that which plans the movement and activates things in your spinal cord, which moves the hands and stuff like that. So it does involve things. But so in a sense. Uh, um, so worshiping does involve your brain, because your brain does the acts of the mind. Your mind, you decide something, you move your hand, the brain carries out the action without you knowing how it does it. So, uh, but and so is it, I don't, I just don't know how what but we worship our mind, so the mind is immaterial, but and so we think with our mind, and I don't believe that is, we're not our mind isn't our physical brain. That's what I was trying to convey. That evolutionarily we are just the brain, the mind is just an epiphenomenon of brain activity. That's which I don't believe is true. Okay. Um, and things like that. But when even when you worship God, you're going to have emotions and things like that, and you move your body. It's going to activate the brain, and you think you, people think you think something, or you sing a song. It's going to activate circuits in your brain because you're producing speech if you're singing and stuff like that. So there's all these things. I don't think yeah, but it just I don't we worship God in spirit and truth, and I don't think it needs to really go into sort of the finer details of trying to split these things, because we don't know what the mind is, and I don't think there is a full answer. But I think you have to realize that our mind causes actions in the brain. Okay, and so even if you worship God, you pray to God, you're going to activate brain processes, because when you speak, your motor cortex will be activated, and things like that, to move your lips and your vocal cords and things like that. And so, yeah, it's sort of, yeah. Anyhow, I don't know how to answer that sort of questions any more than that.
No, I think you did good. It's a very thought-provoking program and topic, Peter, and you're definitely an authority on this issue. Michael says, great interview. So lots of positive feedback on today's program here, Peter. So here's another one, $50 super chat. So again, some real love and support for today's program. Appreciate it, 21st Century Mind. And so he's wondering this, Peter. In 2 Kings, the Lord opened the eyes of Elisha's servant and saw spiritual realities beyond the physical world. Would you agree that brain physics was intrinsic in this experience to process spiritual information? For example, the visual cortex and hippocampus.
Well, I think it's sort of how God wants to relay this information to you. It's like when God talks to you. It's probably not. It's just more like the person is awake and God is kind of basically. Uh, it's a question that I guess the questions that some of vision, whether it's actually a physical event that you're shown actually the physical manifestation there, or whether God is activating in your brain something that isn't there, but you're seeing it like in a dream. Your brain can be activated and stuff like that, or but the events may not be real, but God is communicating through dreams and stuff, which seem as real, because essentially the brain is just reconstruct the brain when in real life. When we get signals, that it's just the brain is basically interpreting signals, say the visual system interpreting signals from the visual system to make sense of it, and so that's why dreams can be so realistic, because whether it taps into the same circuits or not, but it's just if God taps into your brain, or just the brain is creating all these things, it's sort of maybe touching on the same sort of processes. And so now, if it's showing something that is actually spiritual reality beyond it, say basically the real, say angels. I don't know, can't remember what the actual thing was. Is seeing all these angels out there, and if they actually are there, and they're not actually visible to normal people, then I don't know what mechanism God used to show this experience to Elisha's servant. I just sort of um, I think I think I think that those spiritual obviously they seem to be um, angels I guess they can be spiritual beings, but also if they can physically manifest, it's sort of yeah, it's I guess it's a bit, yeah, it's sort of it's a difficult question, but I think that it probably goes beyond the physical world, and I think that
Right.
and in that sense, I believe I believe there are, um, there are things in this world which are not just can't explain by the physical, but how it actually works in your brain. It's just I can't. I don't really know. But certainly it could. It depends if God actually. Yeah, it just depends on whether if no one else can see what the servant sees, then it may be that God has to manipulates the person's brain to see it, the processes, or it's a completely spiritual thing. I don't know. It's very very difficult questions.
Yeah.
So it's a good questions, but I don't know. I don't. I'm not really. I did. I kind of probably more comfortable to answer questions more at the organic level rather than sort of questions which I don't want to which are at this level. But anyhow.
Well, I think you're doing a fantastic job and you're giving us some thought provoking answers, which is important when it comes to these shows. We want to get people thinking. And with the critics, we want to plant some seeds. And so this comes in from Lorraine Drosophila. And she is wondering, why do all species of hominidae share the same cortex white space order, including gibbon? So I'm assuming she's including humans, everything in the human line, plus the great apes.
I'm not exactly sure when you say the same cortex white space, whether you're talking about the white matter between the cerebral cortex and the diencephalon. There's a lot of white matter there. The the white matter fibers there. I'm not really sure, but the thing is, God didn't need to reinvent every. If He created a total different thing for every animal, He can use the same design plans to create things. Um, so if you're creating a physical brain, why could He not use the same sort of nerve fibers? I mean, it's no, but I don't. I'm not sure what I understand the question to be to be honest, because it's just the same cortex white space. I just don't understand the question.
To
I'm
me,
not sure what she means by same cortex white space. Um, the cortex is the grey matter. The white space, whether you're talking about the white matter between the cortex and the diencephalon or the basal ganglia or whatever. I'm just not sure. Yeah,
it sounds more like a common design feature.
yeah,
I mean,
yeah,
if we have
yeah,
similarities and share
yeah,
aspects or systems with
yeah,
other creatures, that's
yeah,
good, especially if it's
yeah.
functional.
It's the same. Why would you need to? What you know? What? Why is it? Why just think about it? Is it why would you need to create something totally different from everything just so that
Mhm.
in a 20 or 21st century, so that people who didn't want to believe in God can be to the basically so that evolution to defy evolution, he had to have to create everything differently. You know, I don't know. I don't know. You know, I don't know that God thinks in those terms. He's thinking if people he says these invisible attributes are made clear to everyone. I'm sort of paraphrasing Romans, but people are basically without excuse in terms of if they deny the evidence of God through creation. So basically, because you find similarities and stuff, it's not an excuse to deny there's a creator of things. And what I indicate is that doesn't matter where you start. The waiting time for all this information to get in there, it doesn't really matter. We just start with bacteria. It's not gonna happen. You know, it's and things like that. So I don't know why you would have to create everything totally differently just to please just so that no one would believe in evolution. You know, I think there's spiritual issues involved, and that and if people are open to God, God searches people's hearts, and if you're open to God, want to believe in Him, want to know Him, He will meet you. But if you don't want God, then it doesn't matter what you say. Even Jesus said, if you don't believe the books of Moses, they won't believe in if you rise from the dead. So no amount of evidence will ever prove, so I'm not presenting, I'm not trying to present people who, if they close their minds and, I'm not, that's not I'm aiming at. I'm aiming at people who still have an open mind, who want to know God, who are like me, who wanted the evidence to believe in God, or not know that God hasn't been disproved by science and things like that. That's what I'm trying to reach a show. If people hate God, or in fact, how can you hate if something doesn't exist? So, but if people don't want to believe in God, then that's up to them. They're free to do that. Um, you know, yeah.
Well, I think that's a great response. Peter, no human engineer, no good human designer goes back to the drawing board for every single new feature. They reuse
Yeah.
features, especially if it's good
Yeah.
designs. And
Mm.
so, for example, there's a critic here, and I would put the homology or similarity challenge back on the evolutionists, because here's an evolutionist saying, here they go again, sticking to common design when cornered. Not cornered at all, because my question to the evolutionists, did they ever predict homology? And from my understanding, they did not predict or expect as much similarity that we find today, Peter, because if evolution's true, think about it. We've been accumulating mutations for millions and millions of years. Those mutations should disrupt these homologous patterns. That signature should be more or less
Yeah.
wiped away if evolution's true. The fact that we find homologous patterns in the biological world screams and points to design, Peter.
Yeah. Yeah. Well, you like I say, you can't. If people want not to believe in God, then they can. But in the end, Romans says they don't have an excuse. The evidence is
Right.
there. And the thing is, if you look at the brain, and you and I don't know what, if you created, if you have, say you had create different car versions of cars, does that mean that uh, they still have a designer behind it? There's still design behind the car, even if they
Mm-hmm.
look similar. And in this instance, they may have different designers, but they all use the plan and stuff like that. So I just sort of don't see the question to ask is how could you go from one to the other? And that's the
Yeah.
problem. You can't just you just sort of, it's just
How do
a
these
story.
systems evolve in the first place? Like you've
Yeah,
pointed to the waiting time problem, this
yeah.
significant problem in terms of accumulating truly beneficial mutations that then have to become fixed or stuck in
Yeah,
place in these
and you can only
populations.
do so many in parallel. The problem is, if you the latest evidence, if there's, say it's 5%, well, it doesn't mean if 1%, but say the 5% difference between human and chimps, that's 150 million base pair differences. So from the alleged common ancestor between human and chimps, that means 75 million base pair differences has supposedly evolved in humans. But a lot of the calculations show that at the most 1,000 could be fixed in the population. So if that's the case, then all the rest, if evolution was the case, if not evolution, but if the if it had been 6 million year separations, all those changes would have to accumulate by just random changes, which would be which means you'd already have mutation or meltdown, because they wouldn't be fixed. They would just be random changes. And now we know that most of the genome is functional, and so most of the mutations are harmful, and but not most of them are maybe a little bit harmful, which is good in the short term, but bad in the long term, because as they accumulate, you get air catastrophe, and the genome just would collapse upon itself. That it hasn't happened yet indicates we're not millions of years old, you know. But you're just going to go in circle. If people don't want to believe it, they're going to find anything to hold on to. But in the end, that, yeah, it's yeah, they can believe what they want. If they want to reject it, basically people don't want evidence of God. Then what can you do? It's sad, but what can you do?
It's sad, but in the end, every knee shall bow. Isn't that right? To the
That's
authority
true. Yeah.
of Jesus Christ. So, Peter, another excellent show with you. Time always flies by. I feel like I can talk to you about these topics and points all day. So, hopefully, we'll have to do maybe a follow up in the future, because I do have a feeling we're going to get a lot of questions and comments after the fact, because it really is, Peter, a fascinating topic. So, I appreciate you, brother, recommending it as a follow up to our series that we did on human evolution and the fossil record previously. So, Peter, I want to hand it to you in case you had any final words, final thoughts, anything you wanted to leave our audience with.
No, well, look, I think the reason I do this is not. I mean, I could probably have better things to do, but my goal is to that people to see that God does exist, that there
Amen.
is hope. And if you look in the world today, particularly young people, there seems a lot of confusion. A lot of things are changing. So culture, society is changing so rapidly. There's no anchor anymore. But God, through Jesus Christ, who died on the cross for you, for your sins, is an anchor, and He's offering you eternal life. And all you have to do is believe in Him. And it says, believe in you, confess your mouth, Jesus is Lord, and believe in your heart that He rose from the dead, and you will be saved. You don't have to work for it, and you can have eternal life and have hope in this world and walk with God, and He can be your companion, and that is real hope. This world offers no hope, and I'm afraid long-term hope and beyond this world, and we don't know how long we have on this earth, and so that's my reason for doing it. Not, I don't even understand why the hardcore evolutionist atheists bother with this, because if I really believe their stuff, what's the point? It'd be all pointless anyhow. Why try to rebut something if you don't believe it's true? Why what do you gain by trying to dismantle it? There's nothing to be gained. You're just wasting your time. I think that so even in the hardened atheists, there's probably some worry there that they may be wrong, and they are wrong. I can guarantee you that you are wrong. That God does exist. I know. I can't share, but I know from personal experience, God is real, and Jesus Christ is real, and he's holding out His hand to anyone who's not sure. All you got to do is ask Him. But if you don't want Him, He'll leave you alone. But if you want to believe in Him, then he'll meet you halfway. You have to make the effort. So that's what I would encourage. That's what I would encourage people with.
Amen. That's the best way to end any program, Peter, my brother. Our hope is not in this world. Our hope is in heaven. This world is crumbling. We live in a fallen world, and so our hope is truly with the Lord Jesus Christ. And the Bible says the fruit of the righteous is a tree of life, and he that winneth souls is wise. So Peter, you're very wise, and I appreciate your work. I appreciate your time.
Thank you.
God bless you, Peter. And I want you to enjoy the rest of your day. I know it's early for you. We're on different time zones. To the audience, thank you so much for tuning in. I appreciate the support. I appreciate the questions, and really just all the positive feedback for these two video presentations. Okay. God bless all, and Standing for Truth is out.